Hey, Hammy here, coming at you today with the video for section 11.3, in which we look at Mendelian inheritance and some of the patterns we see in human diseases that are passed from generation to generation. One of the first things we want to look at is where is that genetic disorder located? Okay, they're inherited from your parents. Okay, what chromosome, where is that gene located at? Because depending on what chromosome, whether it is an autosome, okay, or a sex chromosome, makes a difference in the pattern of inheritance. Okay, because the autosomes, you would see no difference between males and females, whereas if it's sex-linked, let's say it's a disorder on the X chromosome, then you start to see differences in like color blindness that may show up more in males than females. So that affects the patterns of that inheritance. Whereas autosome, you'll get equal chances for males and females because it's on chromosomes 1 through 22. The second thing we look at is that disorder or is that mutation dominant or recessive? Because that affects the pattern we see and how that trait is passed on through the generations. If it's a dominant disorder, okay, remember you can have homozygous dominant or heterozygous dominant. They will both have the disorder. And if you're homozygous recessive, you will not be showing the trait. Whereas if the mutation is recessive, okay, the homozygous and heterozygous dominant will not have the disorder, and you need to have that homozygous recessive genotype in order to have the disorder. So if we look at some examples here, first off, autosomal recessive. Okay, if the trait is recessive, okay, we use something that we call a pedigree to represent how that is passed down through family history. So autosomal recessive, if it's shaded in, okay, circles represent female, squares represent male, hence the symbols here and here. And if it's shaded in, that means they are showing the condition. So here we see an affected female married to an unaffected male. Now notice this is a blank or a question mark because we're not sure of that genotype. We can't tell from the pedigree. Okay, they have kids. Their kids are unaffected. They have 2 daughters, daughter 1, daughter 2. Okay, they both get married. And then here we see an example of where the dangers of inbreeding. Okay, so you've got 2 first cousins that get married. And notice they are both heterozygous, or we would call them carriers of the trait. And now there is a greater incidence of, notice how much it shows up a couple generations later because of that inbreeding. You'll notice with autosomal recessive pedigrees or recessive traits, most affected children have unaffected parents. So unaffected. Parents are just carriers. Carriers have heterozygotes are unaffected, do not show the trait. 2 affected parents, okay, if you're crossing little a with little a, all of their children will be affected because that's they pass on the trait 100%. Close relatives, this is the inbreeding we're talking about, have a greater chance for these recessive alleles to come back together. And we see that not only in humans, but in the animal kingdom. If you look at purebred dogs and some things like that, with some of the traits that they struggle with, like hip dysplasia, cataracts, some of those health problems that come from the very, very close lines of breeding. And again, because it's autosomal, males and females are affected equally. So you should be able to recognize this pattern as autosomal recessive. Males and females, unaffected parents can have affected offspring. 2 affected parents will have all of their kids will have it as well. When we look at an autosomal dominant pedigree, you see a little bit different here. Okay, so we have 2 affected parents. Okay, and you see a much higher incidence of affected offspring between those two parents, because there's a greater chance you only need one dominant allele to have the trait. So affected children will usually have affected parents. If you're a heterozygote, you are carrying that dominant gene, you will have the trait. Two unaffected parents, kind of opposite of the autosomal recessive, unaffected parents will not have an affected child because they can only pass on the recessive allele, and both males and females are equal frequency. It shows up equally in both. Some examples of these traits that we see in humans of autosomal recessive patterns: methylhemoglobinema, okay, which is, it's kind of a relatively harmless disorder. You can take some medication. It's where hemoglobin sometimes breaks down. Hemoglobin that carries oxygen in your blood will break down into this methemoglobin, which has a bluish color. And if I switch, if I go to the next slide here, it'll show you a picture. Okay, and so this bluish color here, you see normal. Here you see that bluish color in the joints and under the fingernails and stuff. That bluish color shows up. It's, again, relatively harmless, but, sort of kind of weird in how that shows up. Cystic fibrosis is one of the most common autosomal recessive patterns in Caucasians. They say about 1 in 20 Caucasians are carriers, and it's about 1 in every 20,000 births. And it comes from the defective CFTR gene, which does not let chlorine, these chloride ion channels do not work properly. And so the mucus isn't thinned out. So water doesn't follow. And so if there's no water, the mucus is really thick. And so they have often trouble with digestion and also breathing because the mucus will build up in the lungs. They often have these thumper vests that will help them cough up that mucus. They'll often be very skinny and have trouble getting nutrients because the mucus that lines our intestinal tracts kind of blocks the absorption. It's so thick it blocks the absorption of the nutrients in the intestine. And so a lot of us are kind of familiar or may know someone with cystic fibrosis. It's kind of said the life expectancy is usually not the best with that, is not super long past teenage years and so forth, because of just the complications, lung transplants, that kind of stuff that are needed because of that disease or disorder. This is just kind of a slide showing the defective chloride ion channel right here that's not letting the chloride ions in here. You have chloride ions that also the water will stay there. And again, we think of osmosis, diffusion, hypertonic, isotonic, hypotonic. The water will stay there as well because of the chloride ions inside the cell. And so out here where the mucus is produced, if the water does not follow the chloride ions, the mucus, instead of getting watered down, stays real thick and sticky, and again affects the lungs and the digestive system mainly. Some of the autosomal dominant patterns or diseases you may hear about. One of the common ones we often hear about is Huntington's disease, which is a kind of the nervous system. And again, it's interesting. We talk in class a little bit about, because it doesn't develop until later in life, and it's often lethal. So it doesn't develop to your 40s, 50s, or 60s. Those people have often got married, had kids. And so are there genetic tests, or would you do the genetic test? If your parent has Huntington's, they probably have one of those dominant alleles, which means you have a 50% chance of inheriting that. Would you want to know, or wouldn't you? Because you could have, it's a 50% chance you're unaffected or 50% chance you got that allele. Another one, osteogenesis imperfecta, is characterized by weak, brittle bones. It's just not enough collagen. Not enough collagen is produced, and the bones become very brittle. There is the one Viking your book talks about, Ivan the something, it would just break bones so easily they'd carry him into battle on his shield. And looking back at some of the descriptions, we're like, did he have this? We often see this with young children who are in the ER a lot with broken bones. With broken bones, that might be clue that into it. It is treatable with drugs to make that collagen to build bone mass, but it's a lifelong kind of thing of taking that. Hereditary spherocytosis is a mutation in the anchorin 1 gene causes the blood cells, again, to become too round. Instead of flat discs, they become more spherical, and they burst easy and can cause complications as well.